Hi, my name is Sarah Kleem and I hope you learned something from this video. Today we are going to talk about CRISPR. CRISPR, in short, is a protein that would be able to cut out mutated DNA and replace it with non-mutated genes using Cas9, another protein. This protein would be able to correct human DNA, basically eradicating genetic diseases such as Huntington's chorea and Tay-Sachs. You would think that this whole project would begin in a lab from Harvard, Yale, Princeton, or some other Ivy League school, but it didn't. It actually started with yogurt. Yep, yogurt. Dairy companies were trying to figure out why some yogurt they had made had a tang to it while others did not. So they hired some scientists to analyze their yogurt and they found out it was due to a virus. Viruses are pieces of genetic material, RNA, that alter the DNA in an organism. Since they are not alive, antibiotics and other medication have no effect against it. A virus would enter the bacteria in the yogurt, altering its functions, causing that tang. But some yogurt didn't have that tang. So how is the bacteria able to get rid of that virus? This led to the huge discovery that bacteria had their own defense system similar to our killer T cells. Some bacteria had a protein that could detect foreign genetic material in the organism, capture it, and then cut it up into many pieces, rendering the genetic material useless. Eventually, scientists like Yoshizumi Ishino, Ruud Janssen, Francis Mochik picked it up and began to modify this protein to work in humans, calling it clustered regularly interspaced short palindromic repeats, or CRISPR for short. Now going into how CRISPR works, it is important to note three key parts: helicase, RNA, and Cas9. CRISPR is programmed to find a certain genetic sequence, or RNA, within the DNA of its organism. Helicase is able to separate the two strands of DNA, allowing the RNA to pair up with it, reading its genetic sequence. If the RNA matches up with the sequence, then it is a targeted gene. If not, the protein moves on to read the next sequences. If it is a match, CRISPR is able to snip that gene out, rendering that sequence useless. This is where Cas9 comes in. Cas9 is a protein that carries the correct genetic sequence. Once CRISPR cuts up the mutated one, Cas9 is able to put in the corrected version. CRISPR then leaves and the DNA strands bind once again, but this time unmutated. CRISPR would allow for momentous breakthroughs in the medical field, especially with genetic diseases. It would also open up a much uglier door. CRISPR would reveal many ethical issues pertaining to how far you could change somebody. People could use CRISPR to design their children with specific traits. Parents could essentially create their perfect child. Governments could use it for genetic warfare. For as many good outcomes CRISPR brings, it also brings dark and dismal opportunities for mankind. But overall, CRISPR is at the front of the STEM world. This affects everyone and everything, ranging from humans, plants, and even bacteria. All that's left now is to see how we as humans decide to use it.